Thank you, Ruth and Huma. To be frank, I feel a little emotional because this is like the third Young Forester meeting that I am here attending, and probably the last one because I might not be here next year unless organizers invite me and pay my trips and everything. So to come from Germany, then I might come. Otherwise, it will be very hard to join this meeting next year. Yeah. Imagine a forest. When you imagine a forest, what parts of the forest are you imagining? Are these trees, maybe small plants, shrubs, maybe grass, maybe insects, maybe some wild animals? Is there anybody who thought about microorganisms living in these forests? Even though we know that they are very rich with microorganisms and there is a huge biodiversity living in these forests, but we never think about them because we cannot see them. That's why they are called microorganisms. And because we cannot see them, we think that they are not important. So today I am going to talk about not only that they are important, but also some interactions that are going on inside these big forests and that we cannot see. And we only see when there is something catastrophic happening or an effect. For example, here in the photo you can see that this forest is diseased, and so a lot of trees are dying. So then foresters immediately realize, oh, we have to do something about it. There is something going on with this forest. I will talk about a disease, basically, which is called chestnut blight. It's caused by a fungus. It is a pathogenic fungus that basically infects the chestnut trees, and it is able to kill the tree. However, there is another interaction going on in this story. This is with the virus. So there is a virus that is able to infect the fungus, the pathogen. And what happens when the virus infects this fungus is it decreases its ability to cause disease. So when the fungus and virus both are present in a tree, the tree doesn't die. But if the fungus only is present, the tree might die. And we saw this, for example, in North America, that almost 4 billion trees died because of this one disease. So you can imagine it's a very disastrous disease. The viruses can spread themselves from one organism to another organism. We call organisms strain in our sense. So when one strain has virus, so the virus here is represented by these green dots. When one organism got closer to another, it is able to fuse with other organism and transfer these kind of viruses, okay? So yeah, fungi are doing these kind of processes like all the time, whenever they come in contact with other organisms. But here is an interesting thing. So would you go and make a friend just because this person belongs to the same species? You say, okay, this person is Homo sapiens, so I'm going to make a friend. No, right? You say, no, I have to have some common interest. I like football, so does this person like football? I like, I don't know, cars, does this person like cars as well? So I have some, let's say, criteria that I'm like, okay, if this person has all of these criteria, then I'm going to be friend with this person. The same way, this fungus also have these some criteria. In this case, it's genes. So if there are 6 genes, and if all the genes are the same, it is going to be compatible. We call it compatible, so that they can come together and fuse. Here you can see when the 2 fungi are compatible, this is what happens. So they completely fuse with each other, and the viruses can successfully transfer. But in all other cases, you can see that they do not combine. So there is this line, they stay away. They say, okay, I don't want to be friends with you. Yeah, why is this process important? Because if they don't combine, the virus cannot spread from one to the other. So the objective, so why this is important, you already realized that this virus we can use that has a lot of potential. We can use this virus to control our disease. But on the other side, the fungus has kind of this defense mechanism that it doesn't fuse with each other. So what we want to do, we want to know what kind of fungi are present, number one, but also what kind of viruses can we use to better control this disease. So this was the objective of this study. For this purpose, we sampled in Cantabria region, which is this part of Spain, northern Spain, and we sampled like different forests. Only one type of the fungus was found, which was very interesting. So to put things in perspective, I did similar study in one small part of Italy. There we found 23 different types, which is very high compared to this. So yeah, this was very strange that only one type is present. But it's also good because now we can use this type of fungus to control the disease in the area. Then here we also use some other genetic markers just to see if the diversity of the fungus is real, or if for example, no diversity of the fungus is real, or is there diversity when in the other parts of the genes, so like the DNA. The next part was to detect virus, to see if the virus is present or not. Normally in the literature, there were two methods being used. So one method was to see if the fungus looks different. So we know that when the virus is present in the fungus looks white. But when the virus is not present, it looks like orange, like this. But we had a lot of isolates that looked like in the middle, so we didn't really know if they have virus or not. So it was hard to tell just by looking at the fungus. So a lot of people were doing PCRs. So this is like just a photo of gel electrophoresis. So they were just doing PCR methods. And during COVID, you realize that doing PCR of a virus was a very complicated process. It took, if you did a PCR test, it took maybe 2 days to get the results. So it can be very expensive and a time-taking process. Therefore, there was no method to detect viruses like immediately, rapid test. There was no test like how you saw in the COVID times that there was this lateral flow test. You could just buy it from pharmacy and just test at home. So I wanted to develop a test that we can use to detect viruses that are present inside the fungus. So we developed a similar test to screen a large number of fungal species, fungal strains. Then we sequenced all of the viruses that we found. All of the viruses that we found were sequenced, and here this looks very complicated, but it's not. The colors represent the place where the fungus was sampled, and every one circle represents one type of virus. So what you can see here is that we found a huge diversity of viruses. You can call them variants. Like, for example, in the COVID time, you were also hearing about Omicron and all other variants. So we found a huge diversity of these variants. But I wanted to know which virus will be the best to use as biocontrol. And for this method, I did one analysis. This analysis basically was based on finding if the mutations that are happening will affect the protein that it is going to produce, the virus is going to produce. And this analysis showed some viruses that were under positive selection, how we call it. Positive selection is basically that the virus has attained more functional or better functioning proteins. So this means that for example, the Omicron strain was better able to disseminate itself, so it was spreading more fastly, right? So the same thing, I wanted to analyze which viruses could be better to use as biocontrol agents. What can we conclude from this study is that we identified which fungal viral combinations can we use, so can be better to control the disease. We identified, for example, EU1 type of fungus is better for biocontrol in Cantabria. Then we also identified which viruses can be used for biocontrol methods. But also we developed a new method to detect fungal viruses that can fasten up this process of detecting viruses in the field. And especially, for example, if the foresters are going to the field, or they might not have possibility of having a lab where they can do PCR methods. So we want to develop a lateral flow where they can immediately go to a forest and take a little part of this diseased part and say, okay, there is a virus in this fungus or no. We also came to know about where the virus is present and which areas the virus is not present, so that we can apply biocontrol in these areas and focus only in these specific areas. One of the paper is published and one is under production. Thank you very much for your attention.